Hi, I'm Stuart Willis of the Marine Genomics Lab at Texas A&M University in Corpus Christi. This video describes the project that we're presenting through experiment.com. Over the next few minutes, I'm going to tell you about the patterns that we've discovered through our research and how we can use them to ask really insightful questions about biological diversity.
My colleagues and I study the Amazon. The Amazon is the largest river in the world, discharging more than 55 million gallons per second. That's more fresh water than the next 7 largest rivers combined. But the Amazon region is extraordinary not only for its size, but also for its spectacular biological diversity. The tropical regions of South and Central America are home to fishes that together make up around half of all vertebrate species on Earth, and the Amazon region contains the majority of these species. But this, of course, begs the question why? Why are there so many species in the Amazon? One major reason is likely the great variety of habitats in the Amazon. For fish, the biggest difference in habitat types appears with the comparison of white and black waters. White waters flow mostly from the mountains, carrying lots of silt and nutrients, having a pH close to neutral, and a high dissolved oxygen concentration. Black waters, on the other hand, flow from very old and sandy soils, contain few nutrients but a high content of organic acids, often have a very low pH and a low dissolved oxygen concentration. One of the best places to see how different these water types are is called O Encontro das Aguas, or Meeting of the Waters. The most famous of these is near Manaus, in Brazil, where the Solimões River, a whitewater river flowing from the Andes, meets the Negro River, an extreme blackwater river flowing from the north, and, according to Brazilians, the two combine to form the Amazon River. Where these two tributary rivers meet, the waters are unable to mix for miles downstream because of the extreme differences in water chemistry. For fish, black and white waters pose very different habitats and must require a considerable range of adaptations for living in one versus the other. It should come as no surprise, then, that many, if not most, species in the Amazon region are restricted to only one type of water. However, those species that are found in both water types give us the opportunity to study the genetic mechanisms that underlie adaptation to each environment. The trick, however, is finding the right species and in the right environment in order to get the best look at how this happens. For my graduate work, I was interested to know what role hybridization played in the origin and maintenance of species in the Amazon, and I chose to examine this in a group of fishes known commonly as Peacock Bass. Aside from my scientific interest, the major reason I chose to study these fish is because of their importance to communities in the Amazon. Peacock bass are exploited heavily as artisanal and commercial sources of food, and there is a considerable tourism industry focused on recreational fishing for these hard-fighting fishes. But fishing pressure and human degradation of aquatic habitats puts these fish at greater risk every day. Knowing what the species and population structure of peacock bass are gives research managers much more effective means of conserving them. Every additional piece of information we add to the peacock bass puzzle only improves the chances that these fish will be around for the long haul. This research was, it turned out, a labor of love on my part. Despite so much interest in peacock bass, fieldwork in South America is not like fieldwork in the US or Europe, especially as a graduate student, traveling and collecting on a limited budget. In the course of the work, I spent a lot of time sleeping in hammocks, covered in bug bites, and hauling equipment on and off of buses and riverboats all over the Amazon. But in the end, I think, it paid off. because not only did I collect hundreds of samples that give us an unprecedented view of species boundaries and population structure of a group of Amazonian fishes, but I also got to catch a lot of cool fish and see some really beautiful and unique places. And having all of this information gives us the ability to hone in on the really interesting patterns and ask the kind of truly insightful and integrated questions that we couldn't ask without this level of background detail. One such place concerns a hybrid zone between populations of two species that originate from those two very different habitat types that I told you about before, the black and white waters. What you see on this map is the approximate distribution of two species of peacock bass in rivers of northern South America, the butterfly peacock bass in green and the yellow peacock bass in blue. Two things are immediately obvious. One, they are both very widespread species with large range sizes. And two, there is an area where their ranges overlap, and there are areas where they do not. If we plot on top of this the water types of the major rivers in each part of their range, we can also see that both species have populations in both black and white waters. This makes the population genetic structure of these species interesting with regards to our original question about how Amazonian species adapt to these different water types, or why they don't. We also observed that these species hybridize and exchange DNA in a limited part of their range. That's unique and interesting enough on its own, but what was strange about this hybrid zone is in what part of their distribution it's located. Although these two species have a broad overlap in their ranges, the evidence for recent hybridization was only found at the very southern end of the butterfly peacock's distribution, right next to where the yellow peacocks are found separately. To explain what we think is going on, we need to take a closer look, so we're going to zoom in on this portion of the map. So here again we have the distributions of the two species, the water types of the major rivers, and the part of their ranges where we found recent hybrids. To give you some perspective, our natural expectation for a hybrid zone would be that fishes living in the same place would be the ones hybridizing, and therefore show the greatest mixture of DNA. However, based on what we saw in our DNA results, this is not what appears to be happening. First, the yellow and butterfly peacocks in the hybrid zone don't appear to be hybridizing. This is not totally surprising, since they don't hybridize farther north where they occur together either. Instead, what appears to be happening is that yellow peacock bass from whitewater localities are migrating into the lowest reaches of the blackwater rivers and hybridizing with the butterfly peacocks there. To some extent, this is also not surprising. When two closely related species are not found together, they don't have the pressure to develop mechanisms to distinguish each other as mates, something scientists call reinforcement. However, in this case, this lack of discrimination should only apply to the yellow peacocks from whitewater, since both species occur together in the blackwater sites, and the butterfly peacocks should be able to tell the difference between species as potential mates. That being said, males of most species tend to be less choosy than females in general, and will mate with just about anybody that comes along, even females of the wrong species. So, in this scenario, male butterfly peacocks might mate with female yellow peacocks, even if the reverse was not true. And perhaps not surprisingly, based on the DNA evidence, this is what appears to be happening, since hybrids have DNA inherited from female yellow peacocks from whitewater and male butterfly peacocks. However, that being said, these are not transitional or intermediate localities in habitat type. The sites in the hybrid zone are full-on blackwater sites. This means that hybrid individuals, as well as the yellow peacocks in the hybrid zone with parents that came from each water type, would have a mixture of genes adapted for black and white waters. But in the hybrid zone, they are having to cope physiologically with blackwater conditions and compete for food and mates with peacocks that have only the gene variants suited for blackwater. Based on this premise, we hypothesize that there is a cyclic process of selection on these hybrids. Each time hybridization occurs, only those descendants of fishes with the right combinations of gene variants for black water survive and pass on their genes. And thus the populations in the hybrid zone may remain mixed across their genomes except for those genes that adapt them for life in this blackwater habitat. This gives us a really unique opportunity to find those genes and measure the rate of selection upon them. Knowing now about this opportunity, we are eager to develop a new DNA dataset using next-generation sequencing technology and look for those genes that are correlated with water type and under strong selection in the hybrid zone. To do this, we will use a type of data called single nucleotide polymorphisms, or SNPs, and the next-generation sequencers allow us to develop thousands of these markers that are arrayed across the genome. When we compare those markers between populations of each species from black or white waters, and the hybrids between the two species from different water types, we expect to see that some markers show significantly high or low levels of divergence, like you see in the figure below. Once we have identified candidate markers under selection, we can estimate what genes these DNA markers are linked to in fishes with well-characterized genomes. We can then determine what effects those genes have in fish physiology and begin to unravel why some Amazon fishes are able to make the jump between extreme habitats and others are not. The fortunate part is that we already have the samples in hand that we need to take the next step. In the Marine Genomics Lab, we also have all of the equipment necessary to prepare the DNA libraries for SNP datasets. But what we do need is the funds to buy the consumable materials for DNA extraction and library preparation, and the funds to have our DNA libraries sequenced. The basic budget that we present on the project webpage provides for enough materials and sequencing for a single SNP set for approximately 300 individuals. Additional funds would allow us to do more SNP sets for those individuals, increase the density of markers across the genome of these populations, and give us more statistical power to discriminate outlier genes. Please consider making this work possible, and thanks for taking the time to learn about our project. I look forward to hearing from you.
Por isso eu vou na casa dela, ai, ai, falar do meu amor pra ela, vai. Tá me esperando na janela, ai, ai, não sei se vou me segurar. Por isso eu vou na casa dela, ai, ai, falar do meu amor pra ela, vai. Tá me esperando na janela, ai, ai, não sei se vou me segurar.